I don't know if everyone has completed the exam. The deadline is today, and I haven't seen your grades. So, but if you have questions related to the exam, we can retake those next week. Okay. So now we are entering into the second chunk of the course, which is biodiversity. So now we are going to learn more details about the different groups of living organisms on the planet. We will start by the more humble side, the microscopic world. It doesn't mean that it's simpler, it's just than us. It just means that it's different and we need specific tools in order to approach them. So the two groups belong to the largest and more diverse part of the planet. Bacteria and archaea, two large domains, and actually the ones that first arose on the planet, the prokaryotes, okay? Anybody remembers when is the first record of life of prokaryotes on the planet?
3.5 billion years?
Yeah. 3.5 billion years, but now we are not even sure. It might have been earlier than that. Okay? So let's begin, and this and the subsequent lecture probably are going to be of most interest to the people going into the health sciences, either human or veterinary medicine. We are going to be talking about groups that are very important from the health perspective. Okay, so again, prokaryotes, we have discussed already some of the main characteristics. Simple cell organisms, not internal organelles, no compartments. And what is interesting is that you can find it anywhere in the planet. Okay, if you think of any extreme environment, you will find them in there. Okay, so we already mentioned that, but one thing is that they are tiny. Some of them can be big in comparison to what is expected, but they are still microscopic. And most of them are going to have three main morphological shapes. So you have the spherical shape, the rod shape, and then the spiral one. So spirochetes, coccid, and bacilli. Okay, those are the three main forms that you are going to find in prokaryotes, particularly in bacteria. Now, we have discussed many things when we compare prokaryote and eukaryotic cells. Prokaryotic cells are going to be very different to the eukaryotic cells. Eukaryotes usually are going to have cell walls, if they have cell walls, because we humans, for example, don't have. We have a membrane, not a wall. But many others, like plants and fungi, have cellular walls made of cellulose or chitin. But unlike eukaryotes, prokaryotes are going to have a cell wall that is structurally different to eukaryotes, and it's made mostly by what we call peptidoglycan. Okay? So it's basically a mesh of sugars and polymers, polypeptides, so sugars, proteins that are going to form this barrier that protects these microorganisms. We will also learn how archaea and bacteria are similar and different to eukaryotes in a subsequent part of this lecture. Now, one very common and general thing that if you have not learned so far is that scientists classify bacteria in two main groups based on their staining capacity, either gram-positive or gram-negative bacteria. So they use this stain, gram stain, and then we are going to have gram-positive bacteria and then the gram-negative bacteria. And what is the difference? We mentioned that bacteria, unlike eukaryotes, or prokaryotes, unlike eukaryotes, are going to have a different structure in their cell wall and membrane. So gram-positive bacteria are going to have this mesh of peptidoglycan right at the top of the membrane of the cell wall, right? It's exposing the peptidoglycan to the environment. Gram-negative bacteria are going to have much less peptidoglycan, and it's going to be in the middle of a double bilipid layer. Okay, so you will have membrane, peptidoglycan, and then more membrane. So when you stain them, they are going to be stained in a different way. Okay, so you have here a micro picture showing you how they are differently stained. Gram-positive bacteria are darker in color, bluish, more violet, versus gram-negative bacteria that are more pinkish, okay, or pale. Those are two very simple ways to classify bacteria, gram-positive and gram-negative. But that will have a lot of implications in what we can do about those microorganisms from the health perspective. So gram-negative bacteria are going to be usually more resistant to our chemicals than the gram-positive bacteria, okay? Why? Because many of the antibiotics that we have produced will focus on the peptidoglycan. Why is that? Because there is no such chemical in eukaryotic cells. So we are not poisoning ourselves when we are using those chemicals to combat a bacterial infection. Now, one important thing that you will learn as you grow in your career is the different methods and strategies that pathogens or parasites in general have developed in order to combat the immune system and of organisms and the drugs that we have developed with our human technology, with our human medical technology. In the case of bacteria, they usually are going to have this protective coat, okay, that we call the bacterial capsule. It can be present in a more rigid kind of a structure, or it can be present in a loosely, more slimy kind of way. But that protective coat allows the bacteria to really attach to cells in the organisms where they enter and probably create a potential disease for that organism. So it's a protective coat. And that coat is going to have a lot of antigenic properties to avoid the immune system, okay? Now, of course, all organisms on the planet at one point or another during the history of life and of their own lineages are going to face harsh conditions, okay? So when that happens in the case of bacteria, they have developed this strategy where they are going to form endospores or very resistant stages, okay? So what you will have is that the cell, when it starts to feel harsh environmental conditions, is going to unlock a programming to develop this structure, the endospore. And the endospore basically is going to contain a copy of all the genetic material of that organism to keep it safe. It's like a vault, okay? To keep it safe from what? From harsh conditions. And then that endospore is also going to develop a coat. And eventually, if conditions continue to be harsh, basically the prokaryote is going to disintegrate and the remaining thing is going to be the endospore with that protective coating, and depending on the species, that protection can allow this microorganism to survive from days to weeks to months to years, okay? Now, some other structures that are very common to be found in bacteria are these extensions that the technical term is fimbriae, And there are different types of fimbria. So some of them are like sensory organs that will allow the organism to know if it is moving in the right direction in an appropriate environment. Others are going to be called pili or sex pili. Why? Because those are going to be longer extensions that bacteria are going to use to connect with other bacteria and to do the famous horizontal gene transfer, where they are going to exchange genetic material, and we will see why that is important. It's kind of a sexual reproduction in prokaryotes. Questions? No? Okay. So, of course, they are going to have motility, meaning movement. And that property of movement will allow them to have a capacity to avoid harmful environments. So when the organisms are going to sense a toxic environment, meaning we are using some drugs or something in the environment is not right, they have the capacity of detecting that and avoiding it, moving in the opposite direction, okay? The technical term for that is called chemotaxis. And many of these organisms, the motility structure that they are going to have are either cilia or flagella, okay? We will talk about those. So here's a diagram of that flagella that you saw moving in the video, and basically it's just like an engine, right? A molecular engine with a set of proteins attaching to the cell. You can see here, right here, this is the engine, the motor. Then you have a rod, okay, that is going to connect to that extension that is going to produce the movement of the organism, allowing it to have this chemotaxis capacity, okay? Now, one interesting feature is that only half of the proteins in that flagellum are really essential. The others can be removed and the flagella will still be working, but that is like a built-in self-structure in these microorganisms, okay? Okay, here is one point that I didn't mention in previous lectures, but it's very important in terms of evolution. Sometimes we find things like these proteins that they are not really essential, and that applies to many other things in biology. So you think, why they are there? Well, they are a byproduct of selection, selecting certain traits that are useful to coping with current environmental conditions. And along with those traits, those other characteristics that doesn't seem to be really relevant at that moment are carried away, okay? Just because they are a structural attachment. You need them in order for the selected trait to be functional, okay? You can see all that property of in engineering designs, okay? Sometimes we just keep certain things that we can get rid of because they provide some structural integrity to the system. So they are there, not really because they are essential, but because they provide certain type of redundancy, security to the system. That happens in biology all the time, even though that redundancy is not a specific selected trait made by natural selection. So those traits that basically tag along with the selected trait are called exaptations. Okay, that's a technical term in evolution, exaptations. And those exaptations, it might not be relevant under the current environmental conditions, but we don't have foresight in evolution, right? We are not goal-oriented in evolution. So we don't know when something can become useful. And it turns out that throughout evolutionary history, many of those traits that were not the selected traits, but were selected because of some reason, they were attached to the selected trait, eventually they turn out to be highly useful for the survival and higher fitness of the individual. So you never know when those exaptations are going to be used. If you want a closer analogy, for example, when you go to a cathedral, those big cathedrals or churches, and you see these vault shapes or domes, you can see at the corners these triangular shapes, okay? Those triangular shapes are there because they provide solid structure for the dome or the arch that they are forming. But then they are just there and providing nothing else in terms of architecture to the people building those things. But then somebody along the history of building churches and those type of domes thought, okay, instead of making it an empty space, why don't we build something beautiful in there? And then they started to put images and mosaics and things, ornaments with meaning. For what? For whatever religious purpose they were building. Okay? So basically, that structure that was selected for to make a building that is solid was later on co-opted to be used as a canvas to produce something with meaning for whatever purposes that person was using. Okay? So that's an example of an exaptation in architectural terms. So that happens a lot in biology. We have exaptations, meaning not useful right now, but maybe later on is going to be selected by evolution. So are exaptations, that concept, is it clear? Yeah? Okay. We have already mentioned this. Now, despite not having compartments in prokaryotes, some of them have managed to produce certain membrane invaginations that provide features that are useful to do some tasks. So, these specialized membranes are either very good for metabolism, different types of metabolism, like for example, a respiratory membrane. So this is a prokaryote, a bacterium that was able to use oxygen. So one question for you, since you already studied that topic. Throughout the history of life, early Earth had lots of oxygen, or it was anaerobic? Okay, no oxygen in early Earth. And as oxygen started to increase, about how long ago, more or less? No? Who gives more? 1 billion years, 2 billion years, 3 billion years. Oxygen started to increase in Earth's atmosphere about 2.4 billion years. But it was very, very low, okay? So, and the reason why it started to increase is because guys like this, aerobic prokaryotes, started to show up on the planet. And they started to produce the oxygen, okay? That then later on started to appear on the atmosphere. Many, many millions of years ago, even billions, until oxygen started to show up in the atmosphere, okay? And then other type of structure complexity in prokaryotes is thylakoid membranes. And what are these useful for? Photosynthesis. Okay, do you remember? Thylakoid membranes are an structural component of chloroplast. And they are responsible for what? Capturing light, transferring light, and then eventually doing photosynthesis for the production of chemical energy. Okay? So we have these two guys with this type of complexity, prokaryotic complexity, to do two tasks. Okay? Oxygen utilization to produce what? Why do we breathe oxygen? What is it useful for? Say it again?
Is it like 30 ATP?
Yeah, and what is ATP for us? Energy, right? Okay. Now, and then the other one is going to be the chloroplast. Okay? To produce what? Chemical energy by capturing photons coming from the sun. Now, other features that are important about prokaryotes compared to eukaryotes is that they have much less DNA. And the DNA that they have is structurally different, okay? Usually they are going to contain circular chromosomes, as shown in this picture here. All this thread shown in here is just one circular chromosome all along, okay? But in addition to that, prokaryotes are going to have other type of DNA that we eukaryotes don't have, which is a small circular DNA known as plasmids. And those plasmids are located in a region that is not a proper nucleus, but is known as the nucleus. Okay, so there is a region within the prokaryote where we find the DNA or the genetic material, and the name is nucleus. Okay, questions? Yes.
I know it's a little far back, but the difference between the flagella and the fimbria, what's kind of the difference? Because I know one of them is a hair-like structure that kind of helps them stick in between cells, and the flagella is more for mobility.
Yeah, flagella is more for mobility, but cilia can also be used for mobility.
Okay.
There are some differences in terms, it is mostly in size and also in the microtubule structures. I think, I don't know if in this lecture I have a picture like that, or I am confused with another class that I give, but basically they more or less is the same arrangement in the attachment to the cell, but they have different combinations of the motors that are actually moving the cilia or the flagella. But the main distinction is size. Okay?
That's between cilia and flagella.
Cilia and flagella. Fimbria is all of those.
Oh,
Okay, it's just a technical term for all these hair-like extensions that you can find on the organism.
thank you.
Anything else? more seconds. Okay. Yeah, correct answer is A. All right, questions? Yes.
So they contain both chromosomes and plasmids, or just one?
Both chromosomes and
Both.
plasmids.
So they contain both of them?
Yeah, both are DNA. It's just it's just we biologists name it different just for our own convenience, but everything is DNA material, genetic material. We just name them plasmids because it is present in prokaryotes, not in eukaryotes. Is this a smaller circular DNA? Okay. We humans have engineered some kind of plasmids to make them larger, especially for cloning purposes. The technical term for those that we have engineered are cosmids. So we have plasmids, which are the natural versions, and then we have engineering some larger circular DNA that we use for genetic engineering purposes, for cloning mostly. So one example is called cosmids. I think they are also present naturally, but we have engineering some of those. Okay. Other questions? Okay. Now, there are some factors that are going to contribute to the high genetic diversity and also many other features that make the life of medical doctors and veterinarians a hassle. Okay? So, why? Rapid reproduction, short generation times, high mutation rates, and a lot of genetic recombination. So all of that allows for large genetic diversity, and genetic diversity from the perspective of a species is very good because there are more variants in the population that can adapt to changing environmental conditions. Okay? So all that is important because that translates into having nowadays a problem with resistance, bacterial resistance to our antibiotics, and we will mention more on that later on in the lecture. So prokaryotes, usually the classical example of the classical reproductive way is binary fission. So basically they divide into two. One cell divides into exactly another cell, equal cell. So binary fission. Now, mutation rates are quote unquote low, yet because they have short generation times, you can see how these can compound, just like interest rate. In a short time, many of the things can become very common. So we will produce a lot of mutations, a lot of variation, genetic variation, in a short time. Short time in reference to human time, right? We are talking about that bacteria more or less double or reproduce every 30 minutes. Depending on the species, it can be a little bit less or a little bit more, but we are talking that in less than an hour, every single prokaryotic cell is already two. So you can see how that can compound, and if every generation produces certain mutations, then you can do the math, just multiplying the mutation rate by every generation, and then we will have a huge population of new genetic variants, okay? Here what we have is just an example of some researchers using Petri dishes and E. coli bacteria and doing serial transfer of populations here. Daily they would transfer the old bacterial population into a new tube without doing anything, just giving fresh media for the bacteria to grow, okay? And just by doing that, what they detected after several thousands of bacterial generations compared to the original population is what? The population growth rate compared to the ancestral population has increased significantly. And why? Because they have produced a lot of novel variants that allow them to multiply even faster. And this curve, if you dig a little bit more into this group of Richard Lenski, is the PI of this group, then they started to do experiments where they subject the bacteria into selective environments, meaning they would put media where the bacteria was not capable of growing. Let's say it was not the proper food that they can consume, and some of the few individuals that were capable of using that novel food started to reproduce faster. So this growth curve that you are seeing here is even stronger in growth. It goes over here, and that is under selective pressures. Okay, so this is just to show how across generations, even without changing very harsh environmental conditions, the accumulation of novel genetic variants allows this initial bacterial population to grow faster. Okay, just without changing anything else. Now we have the issue of genetic recombination, right? So if bacteria were just reproducing in a binary fission way, they were producing clones, well, we might not be having a hard time with antibiotic resistance because they are clones, right? The only way that they can produce something to defend against our chemicals or anything in the environment would be through the novel mutations. So novel mutations that arise into the population to provide these variants. But no, they do genetic recombination. And genetic recombination is not only the horizontal gene transfer that we are talking, that probably most of you know about. There are three main ways that bacteria have sexual reproduction. Okay? One is called transformation, the other is transduction, and the other is conjugation. Okay? So all those are put together into what we call horizontal gene transfer. Okay, but horizontal gene transfer might mean different things. Transformation, so basically what the cells are doing is like there is floating genetic material in the environment, and then they detect it, and then they eat it and integrate it into the genome. Okay, it's like something like going to a buffet. You have many things around that you might like, and you pick the one that looks the best and you consume it. Okay, something like that happens with bacteria. Of course, there is a lot of nuance into the biochemistry of the process of integrating that environmental DNA into the actual genome of the microbe. Okay. Now, transduction. This is a little bit more elaborate, and it has to do with the pathogens of bacteria. Who are the pathogens of bacteria? We call them bacteriophages. Bacteriophages are viruses that kill bacteria, okay? But what happens to a virus, any virus, if we know, especially retroviruses like HIV, what they do? They come into the cell host, they attach. If they avoid detection, they inject their genetic material. Then the genetic material travels to the genome of that host cell and then inserts itself, okay? And when it inserts itself, then the next replication cycle, okay, of the cell, or even when the virus hijacks the machinery of the cell to replicate DNA, they are going to produce this novel recombinant DNA, okay? Once that novel recombinant DNA is produced, what do viruses do? Those viral particles, now they are recombine, okay? So the host DNA is going to contain that viral genes, okay? So those new viruses are going to be different, not similar to the original virus. Now, once the genetic material is ready, it has to be packaged, okay? So that's the viral capsid, okay? All these proteins, so now the virus, the new viral particles are put together in these capsids, and now the new viruses come out of the cell, of the host cell, and then they are going to infect other new cells. And they are going to inject what? New genetic material, okay? It's not the original viral DNA, but rather a recombined viral DNA. Recombined with what? With the bacterial DNA. And that new combination can transfer genes from one bacterial organism to another bacterial organism that can have genes to protect the bacterium. Against what? Against our drugs. Okay, this is another way to recombine genes between bacteria using an indirect way, using viruses. Now, the most common and known form is the conjugation, where you have this specialized pilus that is long, but it's a tube, okay? So that tube is going to connect two individual bacteria, and then through that tube or tunnel, they are going to exchange genetic material, okay? So that's the third way to conduct horizontal gene transfer. Now, it's not, I want you to keep in mind why we are having such a hard time with resistance to antibiotics in bacteria. They have three different ways to exchange genetic information, okay? Not one, not sexual reproduction only, but three different types of sexual reproduction. So you can see where the problem can be. And in addition to that, they have short generation times, okay? So a lot of mutations can be produced with those type of characteristics in an organism. Now, as I mentioned, it's not like they are just exchanging genetic information in any way. So there is a little bit of nuance. There has to be genetic compatibility, okay? But some of the important things that can happen is the transfer of fragments or genes of DNA that are called F factors. F for the fertility, okay? So that fertility factor is going to be very important for what? Fertility for the bacterium. Fertility means higher fitness. And those fertility genes or fragments of DNA are the ones that contain the genetic information for resistance against antibiotics. And there are two ways in which those F factors can be transferred. It can be present in plasmids, that circular small DNA that is only present in prokaryotes. Okay, you can see here the original cell, they form this tubule, the pilus, to do the horizontal gene transfer. So here you have the F plus or the fertility factor present in the plasmid, and then eventually the plasmid is transferred to the new cell, and now the new cell has that extra genetic information. Now it's resistant. The other way is that the F factor is going to be present in a portion of the large circular chromosome of the bacterium. And again, they form this connecting tube to transfer genetic information, and then that genetic piece of information containing the fertility factor is transferred, and then it is inserted into the main genome of the recipient bacterium, okay? And then again, the bacteria receives extra information, allowing it to be resistant two chemicals. Now, we for sure know that antibiotics kill many bacteria, but there is a lot of resistance, okay? Now, there is a specific name for a plasmid in bacteria that is resistant to mostly any of the chemicals that we have. It's called the R plasmids. And R plasmids is specifically for resistance, okay? So, this is troublesome because it's the plasmid, the genetic information, creating most of the trouble for the pharmaceutical industry at trying to develop successful drugs to protect ourselves against these bugs, okay? Now, the R plasmids are the ones that have genetic information to develop these tubes to conduct the horizontal gene transfer. So it's not just by chance that the tube or the pili is produced. So bacteria having this R plasmid are also biased into the production of this tube that is going to connect to individuals. So you can see again the problem in there. Now, the plasmid is going to have the resistant genes, and it also has the information to say the bacterium, hey, produce the tube that is going to connect to other individuals, okay? So it has been highly selected because now it's not only the genetic information for resistance is present in the population, but in addition to that, that genetic information is also telling the cell to produce as much connections with other individuals into the population. And what does that? It produces more resistant individuals, the transfer of the genetic information to make them resistant. Are there questions? No? Okay. Now, there are different ways that these organisms can acquire the foods, the energy that they need in order to grow and to reproduce. These are the technical terms for the different ways that they can acquire their food and their energy, but basically it is summarized in here, and we are very familiar with all of them. Photoautotrophs is just like plants, right? They use light to produce the chemical energy. Chemoautotrophs is similar to this one, but instead of using light, it uses inorganic chemistry. Especially this is present in organisms where light is not abundant, like for example at the bottom of oceans. What other place we would have chemoautotrophs in early Earth? So what was first? Let me rephrase that question. What do you think was first in early Earth? Chemoautotrophs or photoautotrophs?
Sun.
Say it again? Yes and no. Your reasoning is right because there is the sunlight coming into the planet. But how were the conditions in early Earth? Yeah, it was chaotic, and many of the sunlight was not reaching, especially the turbid waters of the early Earth. Okay, so a lot of the early organisms were chemoautotroph. Okay, and remember, there was also not much oxygen. Okay, so what do plants produce as a byproduct? The oxygen that we breathe. Okay, but there was not much oxygen as a start. Okay, so early Earth was mostly chemistry. Okay, chemoautotroph. And then we move to the place where we are. So we have photoheterotrophs, okay? They use light as an energy source, but they also need to use organic compounds in order to produce their foods. They are not like plants. And then we are here, chemoheterotrophs. So we just consume food in order to produce the energy that we need, okay? So those are the main divisions. Now, in terms of metabolism, so we have what we call obligate aerobes, okay? Obligate anaerobes, and then we have one version that contains both, facultative, okay? So we can have prokaryotes that need oxygen for sure, those that do not need oxygen, and actually oxygen is poisonous for them. And then we have the facultative version, meaning if there is oxygen, they would use oxygen because it's more efficient way to produce energy compared to the anaerobic pathways, metabolic pathways, okay? So nitrogen is very important in terms of metabolism for prokaryotes, okay? Why nitrogen? Because nitrogen is one of the main components in the molecules, the macromolecules, that are going to store the genomic information that is going to be transferred to subsequent generations of organisms. So nitrogen is very important, and prokaryotes, bacteria in particular, are very good at collecting the nitrogen from our atmosphere. But in addition to that, we need them, and we need them heavily, otherwise we would die. Why? Because we cannot use the nitrogen from the gas, okay, from the air that we breathe, okay? We need it in a different form. So bacteria, there is a set of bacteria called nitrogen fixers. So they are going to grab the nitrogen in gas form and fix it into a form that can be used by other organisms, like in the form of nitrates or nitrates, okay? Also in the form of ammonia as a waste material. So now in this form, it is available to other microorganisms and also to other organisms like plants and animals. Now, plants, there are some groups, especially in algae, that have developed a type of cooperation. Why? Because in the same colony of organisms, they are going to have portions of cells specialized in photosynthesis to capture the solar energy and convert it into chemistry, and then they are going to have some specialized cells that are going to fix nitrogen to feed the nitrogen to the other parts of the organism. Those specialized cells are called heterocysts, okay? And this is highly common in filamentous cyanobacteria. See, those cyanobacterium are some of the early prokaryotes that were present in early Earth. The first photosynthesizers in the planet are within this group, okay? Are there any questions? Okay, 10 more seconds. Okay. See? seconds. Alright. Are there any questions? So, one other thing that give humans a headache with bacteria. Many bacteria can form biofilms, okay? And biofilms can be present in any type of environment, even in very arid conditions we have biofilms, okay? So biofilms basically have a we can say two-layer structure. The very top layer is going to act as a protective layer for the other layers inside the biofilm. But in addition to that, it also acts as a channel to bring the nutrients, the food, to the interior portion of the biofilm. Now, this gives biofilms very important characteristics. They are highly resistant to very harsh environments. And the reason, or the consequence for that, is that they can produce a lot of problems for whatever things we build. Okay, so they produce industrial corrosion, so it's always a problem. That's why it's important to always have our devices clean, because otherwise bacteria, biofilms will overgrow whatever we are using and will start to deteriorate it, okay? So medical devices, that's an important one. So we need to keep them clean, and every time is more and more difficult because of biofilms and resistance towards our chemicals to clean our medical devices. Your teeth, very important to keep them clean, to brush daily at least twice, because otherwise you have the plaque and then you have cavities and all the issues that you know. Okay, so all these issues are important in terms of biofilms. Questions? Few more seconds. Okay. Perfect. Questions before we move into the next concept? Okay. So we have already mentioned that prokaryotes at least date back to 3.5 billion years ago, okay? And we can find them all over the place. In any environment that we sample, we will find prokaryotes, okay? But there are some lineages that are going to be present in extreme environments, and we will talk about those in a second. Okay, so when we have conducted genome analysis, what we have found is that we have these two main groups, okay? One is the archaea and the other is the bacteria. Now within the archaea, we still lack a lot of knowledge in terms of the relationships of these different lineages that are listed in here. As you can see here, in this phylogenetic tree, what we have for the archaea is what we call in systematics a polytomy. A polytomy means that we have the common recent ancestor, but we don't have any other information. We know these four groups are related to each other, but we don't know which ones are more closely related to each other or which one is the basal clay. So a polytomy means that we have a monophyletic clay, but we don't have much information, okay? We need more information to really piece apart the relationships of these groups. We will talk more about these groups in a sec. Now, this I have mentioned already, but it's very important that you keep it in your mind. Horizontal gene transfer in its different versions has been very important for the evolution of prokaryotes and even eukaryotes, as we will learn later on. Okay? So, one example of research is that when people look at just a little over 300 bacterial genomes, on average, 75% of all the genomes that they analyzed were a chimera of different things from different bacterial species, or quote unquote what we consider bacterial species. And if you want to have something more solid on the grounds of the importance for evolution of horizontal gene transfer, you just have to look at the human genome. The human genome is a mosaic of archaea, bacteria, and viruses, in particular retroviruses, okay? All right, so bacteria, in general terms, you will have these 5 groups: Protobacteria, Chlamydia, Spirochetes, Cyanobacteria, and gram-positive bacteria. When we are talking about Protobacteria, we are talking about gram-negative bacteria. That includes many different ways of acquiring food and energy, listed in here. One example is this one, that is an autotroph that obtains energy from oxidizing sulfide, okay? So hydrogen sulfide is the chemical that is going to use to produce their energy. The waste material is sulfur, okay? So you can imagine where this organism lives. Can you guess where you would find this type of bacteria? If the waste material is sulfur, where do you think you can find it?
Garbage cans or smelly?
Yeah, garbage cans, but in the natural environment, when you are taking a hike in the forest, usually where do you feel that smell?
Corpse, like an animal corpse.
Yeah, but wetlands, right? Bogs. Yeah. Ponds. Okay, so this smell to sulfur that is not very pleasant, at least not for us. Okay, those are places where you can find this type of bacteria. Now, let's check your knowledge. Gram-negative bacteria. How is the peptidoglycan of this type of bacteria organized on the cell wall?
In between the bilipid layers?
Yes, in between the bilipid layers, okay? Now we have heterotrophic proteobacteria that include many pathogens that are famous for all of us. One is Neisseria gonorrhea that causes this venereal disease known as gonorrhea. Then we have Vibrio cholera that causes cholera, so food poisoning, some type of food poisoning. Then we have Helicobacter pylori, which I think is considered a neglected disease because it's not as damaging and as apparent as many other bacterial diseases from this group, but it produces ulcers in the digestive system. And especially is present in countries where there is no good healthcare system. And it can be very detrimental to children, okay? So it's a serious thing in developing nations, okay? So for those that go into medical school, Helicobacter pylori is going to be a thing that you will commonly find. Now, one thing that is important is, if you remember, we've been talking also about endosymbiosis for the production of the first eukaryotic cells. The mitochondria that we all have in our cells is a descendant from this group, from proteobacteria. Okay? Questions? No? Okay, let's move into chlamydias. So chlamydias also are gram-negative, and one example is this one, Chlamydia trachomatis, and is a common venereal disease, and it's very common around the world, and in the United States it's also fairly common. So it causes this type of urethritis, basically a lot of pain in the urine ducts. So whenever you are peeing and you have this type of infection, it's going to burn and be very, very painful. Okay, so it's very common. There is good treatment for it, so yeah, it's not too much to worry. Now, the other group is spirochetes. Also gram-negative. They are helical. Remember those general morphological types that we discussed at the beginning of the lecture? So spirochetes are that type of helical shape. They are gram-negative, and we have some examples in there. One example is the one that produces the disease known as syphilis, and the other is the famous Lyme disease. The difference between the two is that one is venereal disease, meaning sexually transmitted. The other one is vector-borne disease, transmitted by ticks. Okay, and that one is fairly common in the northeastern United States. Questions? No? Have you heard of Lyme disease? Yes. You know it's transmitted by ticks. Do you know who the reservoirs, the vertebrate reservoirs of those pathogens are? Ticks are the vectors.
Deers.
Oh, deers. Yes, deers. What else?
Dogs.
Dogs might be, yeah, close to peridomestic areas. What other one is important, if you have heard? It's a small mammal.
Ticks.
Ticks are the vectors. I'm talking about vertebrate reservoirs. A small mammal.
Rodents, like rats and stuff.
Yeah, rodents, but in particular among the different reservoirs that where you can find this disease is the black-footed ferret. I think is the common name of that mammal. But some further nuance used by disease ecologists. When biologists, in particular disease ecologists, realize that there is a mast season for oaks, let's say this year turns out to be a huge production of acorns, a mast season for acorns. We know as disease ecologists that we have to prepare for a high prevalence rate of Lyme disease in the next 2 years. The prevalence peak is going to occur in the next 2 years after the mast season of oaks, acorns. Anybody can guess? This is an ecological detective work, but something that you have to learn to develop as medical doctors, especially if you are trying to figure out where the thing came from. Okay? Why could a plant be related to a disease of animals? Because they? No, in this case, poop has nothing to do with the ecology of this example. Acorn is food, right? Food for what? For many animals, right? Including us. Alright? And we mentioned that there are many reservoirs for Lyme disease in the environment. When there is plenty of food, what happens to populations? They increase in numbers, right? Because it's very good season. So everybody has enough food resources, everybody can produce babies, etc., etc. So I mentioned that one of the main reservoirs for Lyme disease is the ferret. So when ferrets have lots of food, are going to produce lots of babies, lots of animals that can be reservoirs for the disease. And then a high population density of ferrets and other reservoirs means, especially in peri-domestic areas, that the contacts between humans and domestic animals and these reservoirs is going to increase. How? Directly or indirectly? Or both?
Both.
Both, but who will be the crucial link for Lyme disease, jumping into humans and domestic animals?
Well, you're saying it comes from the ferrets, then it gets to the ticks, and then that infects.
So the ticks.
Yeah.
More ferrets, more reservoirs, more ticks, and closer to human habitation, more tick bites. More tick bites, more Lyme disease, and boom. And that happens after 2 years during a mass season of oaks. Why? Because the complete life cycle of the Lyme disease in ticks takes 2 years. Okay? So you see now how nicely we connected ecology with disease ecology and with a bacterium that creates a lot of troubles in humans. We will talk more about this type of connections in the last chunk of the course when we deal with ecology. But I just wanted to bring this to your attention. So you have to think interdisciplinarily, okay? Always, especially if you are going to be in clinic, veterinary or human clinic, you will need to figure out where the things are coming from. And for that, you need to develop an interdisciplinary mind, because the answers are not easy to get. You can diagnose a disease, but you need to know the ecology of the disease, okay? All right, other groups, cyanobacteria. This is great because this is the cyanobacteria that was the first endosymbiont to produce what? Oxygen. Very important, right? So once cyanobacteria was present in early Earth, we have oxygen. And then through the endosymbiotic process, we start to produce photosynthetic eukaryotes, right? So this eventually gives rise to what? To plants, for example, more complex oxygen producing organisms, okay? Oh, and just as it says in there, the chloroplasts that we find in plants were derived from bacteria that are descendants from cyanobacteria, okay? Just like mitochondria is a descendant from proteobacteria. This is just a video of filamentous cyanobacteria, so you can see them. They are very common in aquatic environments, and they are very common in the food chain of many different organisms, okay? Gram-positive bacteria. Okay, if we are talking about the capacity of bacteria to resist antibiotics, which one is more susceptible to our antibiotics, the negative or the positive? Gram-negative or gram-positive bacteria?
Gram-positive.
Why? Yeah, and why is that important?
Because antibiotics target that specifically.
Yeah, that is correct, right? That's the reason why gram-positive bacteria are easier to deal with our chemicals, okay? Now, there are different groups within gram-positive bacteria. We have these actinomycetes, which were initially confused with what? How does this name sound? Probably you have heard. It's a complex group of organisms that we will discuss later on. Fungi, okay? So there are some groups of bacteria that produce kind of fruiting bodies that are going to rot organic components, okay? They are within this family of bacteria, and they are very important because they are present in the environment as decomposers. So they have a very important ecological role. If this group of bacteria would be to disappear, then the only other group doing decomposing would be fungi, and they wouldn't manage to recycle all the organic material on their own to restore all the chemicals in the environment. So this is a very important ecological group. Then we have another group of gram-positive Streptomyces, and we have managed to get a lot of our antibiotics out of this bacterial group, including tetracycline, which tetracycline now is mostly useless to treat just using it, any bacterial infection. So we need to use this one with a combination of other things, okay? But we have used this bacterial gram-positive group to produce some chemicals that we use for our own health. Other groups are naming here. So gram-positive also have pathogens. One famous one is the anthrax, okay, listed in here. The other one is botulism. Anybody has heard of botulism? Okay. What can you tell me about botulism?
It's used for cosmetics. Right?
Yes. What else?
You get it from unproperly stored foods like
Yes, exactly. And what is the more likely route to get it into your body when you are managing food?
Fermenting.
Say it again?
Fermenting.
Yeah, fermenting, and that commonly happens after you open a can of food, okay, and you leave it there. When you are eating canned food, make sure that after opening, you store whatever you are not eating, not in the can, but in a glass container. If you leave it there, especially if you don't put it into the freezer or in the fridge, you are very likely to get this thing. And those are very bad news because it's not easy to treat, okay? Questions?
Is it anaerobic or aerobic? So if like you starved of oxygen, you would die, or
That's a good question, and I don't remember. I don't have an answer for that. Yeah, thank you. Other questions? Okay, 10 more seconds. Okay. Let's see. All right. Are there any questions before we move into the other huge group of prokaryotes? No? Okay. Yes.
I just wanted to go back to the, tourism thing. So even if you're conducting something and it's in a low acidic environment, if, let's say the, I don't know, like, the food that you're fermenting is kind of like elevated above the liquid, the acidic liquid, that could still cause botulism, even if the environment is acidic.
I could say that it might. Because everything depends on the structural complexity of the bacteria that we are discussing. In this case, it's particularly focused on botulism, and I am not really sure about the physiology of that microbe. So that would have to do with the type of environment that it prefers. But usually, botulism is going to thrive in fermenting things. And fermenting things, as you just mentioned, is going to increase acidity. Right? So it might be, yeah, I would say yes. Okay. Other questions? No? Okay, I'm going to give you a few minutes to stretch and to go to the restroom before we move into IKEA. And also for you to wake up a little bit.
Notes and had extra information.
Apparently, you know what, I tried to read it, and apparently if you have to use iNaturalist like iOS app, it doesn't have the project feature. So you have to do it online. Like if you have to publish it, if you already published it, and then you go to their website, log in, and then like add it to the project.
Okay, I need to do that.
I also
And they old apps give you like a project section where you can like enter the project that you want to post it on.
It's not human everywhere. Oh, no, yeah.
But like, I don't know if falling. Isn't that cool? Oh my god.
Who was I talking to? Oh, yeah, my gosh.
I was like lifting off and drove from there, just like finding stuff.
Oh.
I know. We were like in nature and stuff.
My house. Oh, it's because I was getting information from home.
The other day where I went to like here, to like closest like conservation park. It's all like
appointments.
I was like, yeah, I was like,
Back and forth.
over there and just like, yeah.
My parents were
My dad goes to this like little outside and like doing stuff. And then like it's just low key like
I
just
live in an apartment complex.
like
People
out
don't tell me.
some in the rocks and stuff.
Incorrect.
And you left
So then
like
when
some wood and they user friendly seats. I found a bunch of like wood stuff today.
The plumbing, like they close it right to my patio.
I was just lifting.
Like it's flooded. So we have to do all maintenance, there's water in the roof, there's water in the walls, mold's gonna grow, it's a whole mess. Like, yeah, once I'm healed, I just wanna keep doing it. I think I
And my sister killed it before I could take a picture. I was like, yes. This was weird.
It's been fun. We're dealing with all this.
Okay, guys, let's resume the lecture. So moving now into Archaea. So the other huge domain of life. And as we have mentioned before, Archaea is a very interesting group because it's sitting in between bacteria and eukarya. Here on this table, you can see that there are some general features of Archaea that are shared with either bacteria or with eukarya, okay? And for example, let's take peptidoglycan is absent in Archaea, present in bacteria, as we have discussed so far. Now, some things that are present and shared between Archaea and eukarya is different polymerase RNAs, meaning RNA enzymes. Whereas bacteria only have one kind, Archaea and eukarya have several of them, okay? Now, other important thing is the type of amino acid that is going to be used as the green light to say somewhere in the genome, okay, you can start translation of the gene in here. You can see that Archaea shares methionine with eukarya, and then bacteria has another green light in the genome to tell the machinery to start replicating the genetic information, okay? So those are some of the characteristics which makes obvious that how everything or all life on this planet is just a mosaic of everything around us. We have been exchanging genetic information for billions of years now, okay? Now within Archaea is where we find the organisms that can survive in extreme environments. So we have the allophiles, meaning they can survive in extreme salty environments. The other ones, thermophiles. Those are the most famous ones, right? So those that are able to survive in extreme high temperature conditions. We have methanogens. Basically, those are obligates anaerobes that are going to produce methane as a byproduct. And of course, we can find it in many different places. One unlikely place is kilometers under the ice in Greenland. So we just discovered those when drilling those ice cores on in the poles, okay? So others that are classical, and that's where the smelly or smell sulfur comes from, is in swamps and marshes. And then in terms of organisms, where do we find them in organisms? We have cattle, termites, and other herbivores. Of course, the most common not common, famous ones, is the cattle that we produce. Okay, so what is one of the main problems that environmentalists talk about with the high production of cattle that we humans have around the world? Is the production of a greenhouse gas, methane. Okay, so that is the main problem in terms of environmental conditions. We have other groups here. I always have a trouble saying this mouthful, but you can read it there. So other allophiles, and then we have other groups, other large clades of archaea that not necessarily are closely related to, for example, the classic allophiles or thermophiles. So what we have here are two groups of archaea that have converged into the same trait, ecological trait, meaning they can resist high temperatures or high salinity or other conditions, okay? And remember, one problem that we have with archaea in terms of phylogenetic relationships is that we don't have much phylogenetic resolution in this large group of archaea. We just know who the basal lineage of archaea is, which is this one over here. That includes many of the extreme allophiles, okay? Now, the other group that we are talking about here is this large clay that is a polytomy. Again, we don't have much resolution in this group. It has this name, TAC. TAC is referring to the four groups or the four lineages that we are considering in this group. Okay, that's the reason that huge clade is called TAC, just referring to the names of the different lineages. Now, one recent discovery is of this archaea group, the Lochiarchaeops. And this one is very important because not only is closely related to this larger group, but it can be actually the sister group to the eukaryotes, meaning the sister group to all the complex life on the planet, all the eukaryotic organisms, either unicellular or multicellular like us. So it is still not placed in current phylogenies. Maybe in the new edition of the textbook that is coming this year, it will be placed in the phylogeny. Okay? Are there any questions? More seconds. Okay. All right. This just to restate something that we have been mentioning a couple times already. Prokaryotes are the base for basically life running on this planet. If they would disappear, there will be a cascading effect leading to basically life disappearing on the planet. And in particular, one of the main roles is prokaryotes acting as decomposers. So they are the recycling machinery for most of the things that die on the planet. Okay? If they are not present, other decomposer groups like fungi are going to have a hard time trying to process all the organic material that just died. Okay? Other important function is nitrogen fixation. Okay, we have mentioned already some groups of bacteria that are very important for this. So why do we need them? Because we cannot use the nitrogen that is in the air. Most complex organisms cannot use them. Okay, so we need nitrogen fixers. And some of the most important nitrogen fixers are bacteria. Okay, so you can see also that there are some differences in bacteria. In these experiments that some researchers did, is that when bacteria was not present, the uptake of nutrients by plants, in this particular case potassium, was significantly lower compared to other plants that were having a different strain of bacteria. Some bacterial strains are better at fixing allowing plants to acquire nutrients than others, but when they are present, they increase the absorption of nutrients by plants, okay? So bacteria is very important for that reason. They are going to fix the nitrogen in the soil, and that nitrogen is going to pass into the food chain that we all need, okay? Are there any questions? Yes.
So when the bacteria is nitrogen fixing, does that mean that it's turning the nitrogen gas into a different type of gas in the atmosphere?
Not a different type of gas, but rather into a different type of more complex molecule in the soil, like nitrates, okay, that can then be used by other organisms, by plants. Basically, what are our plant fertilizers? The things that we buy in Lowe's, Home Depot. So it's food for the plants, okay? Other questions? Okay, so this is what we just mentioned is very close to us because we need the plants to produce our food and the food also for our farm animals. But also bacteria is very important in other communities that we don't see very often. For example, in the hydrothermal vents in the ocean floor. So bacteria right there are basically the base of the food chain. Why? Because they are going to produce the food for all other organisms in those tiny, compressed and compartmentalized ecosystems. They are going to use chemical energy to produce food for the rest of the organisms depending on them. Okay? Other questions? No? Just a brief thing about ecological interactions, which is important. Ecological interactions are under the umbrella term of symbiosis, okay? When you hear symbiosis, it refers to some type of ecological interactions. Now, of course, ecological interactions come in different flavors. Famous ones, mutualism, that is benefit for the organisms interacting with each other. We have commensalism, where one organism gets a benefit and the other doesn't get any benefit or damage. And then parasitism, where one gets a benefit and the other gets damage, okay? So those are like the general types of symbiosis. But one thing that I want you to keep in mind is that these are not fixed boxes in nature. Just because something is a mutualistic interaction right now, it doesn't mean that it cannot turn into a pathogenic interaction in the future. Everything depends on the current environmental conditions. There are experiments and there are a lot of evidence showing how a commonly recognized mutualistic interaction has turned into a more pathogenic type of interaction, okay? Now, other important thing before we move into the next concept is that pathogens are a subgroup of parasites, those that go into the medical field. What is the difference between a parasite and a pathogen? Why do we make the difference? Or how do we categorize a pathogen? Why we say a pathogen is a pathogen? So all pathogens are parasites. Pathogens is a subgroup of parasites. But why do we call them pathogens? Yeah, and what is disease? If I ask you, okay, what is a disease? How do I recognize a disease?
Releases antibodies in the body for activates the immune system. I'm not sure
Okay,
if it's-
there can be an immune reaction, sure, but then what happens after that? If you are a clinician, how do you know someone is sick?
It's an attack on, like, adding on to what he said, it's an attack on not only on the immune system, but also reflects on how you act, so it's you weakening the benefits that you can provide to the environment, maybe? Like, you can
Okay.
understand and see someone sick because they're not at their best.
How do you know someone is sick?
Symptomatic.
Huh?
They're symptomatic.
Symptoms. That's the key word, right? So
Parasites.
a pathogen, not all parasites cause disease. Okay? So parasites can have a negative impact on the host that they are exploiting. Yet, that doesn't necessarily lead to losing homeostasis. Losing homeostasis means being sick. Okay? And pathogens, unlike the rest of parasites, produce disease. Disease means symptoms. Okay? So you can recognize that someone is sick. Okay? Yet, is that, as a medical doctor, future medical doctors, what I just said is bulletproof? So that's the only way that, that's the best way that you can diagnose a pathogen producing disease? So no, why? Okay, yeah.
They're both symptomatic regardless, so it's kind of hard to distinguish.
Say it again?
They both cause symptoms in patients, so it's hard to distinguish or differentiate what causes or what categorizes as a parasite and what categorizes as a virus. I mean, maybe there are different ways to treat them, is what you can distinguish them as. I mean, I'm sure there's different ways to treat parasites compared to treating viruses.
Not really. Yeah, the reasoning you are using is fine, but I think I am aiming to a different topic. Let's take as an example the COVID pandemic that we all went through. Okay, so did all patients that got infected by COVID present symptoms? No, right? So they were basically not sick, right? They didn't even know, many people didn't even know that they got the virus because the virus didn't do anything to them. So the question is, were they sick or were they not sick?
They're infected.
Okay, that's a good point. They were infected, okay, but they didn't have symptoms, okay? So the reason I'm digging deeper and deeper and deeper into this is for you to see all the nuance that you are going to face in the future when you are dealing with clinics, okay? With making decisions about symptoms, pathogens, what decisions to make, what decisions not to make, when do you need to dig further to gather more information before you make a decision, or you prescribe something, okay? Yes.
Can it be like when people are asymptomatic but they can still be contagious?
That's another nuance, right? In the COVID, were all individuals that were infected with COVID but were asymptomatic were also infectious to others?
Only if they're symptomatic, right? If they produce like sneezing or type of saliva, or dangerous saliva and secrete that contain the virus, then it can infect other people.
Okay, but all of those asymptomatic infected people were contagious to others?
I see like some of them, if not most of them. Yes.
Well, some of them,
It's
right?
niche, it's niche.
Some people were infected, were asymptomatic, and they were dead ends to the virus. Others that were, yes?
Should it be the same, like, when people have a parasite, like maybe people, like if you're swimming and you get a leech and that leech doesn't really carry a disease, or like how maybe if dogs get ticks, but the ticks aren't carrying disease, but the dogs have enough ticks that it's causing the dog to get sick still, so a parasite can still cause symptoms, but not necessarily be considered a pathogen.
Yes. Yes, because in that case, the tick is a parasite, it's feeding on blood, but when the intensity of that parasite is too high, well, basically they are drawing blood and blood and blood, so we have just enough blood, right? So that is indirectly going to cause the loss of homeostasis of that individual that has a heavy infection, which is going to lead to a host of many different symptoms that then you can call a disease. Okay? So you see how complex things are. It's not as simple as sometimes we might like to think. So in biology, we like to put things in boxes because life is easier for us in that way. But those boxes are artificial. Okay? So you have to learn to see the blurriness in the boundaries of the boxes, in the concepts that you are going to learn throughout your career.
It's like you said, like it's a niche, right? It just depends on the virus. I mean, some viruses they can spread asymptomatically. Some viruses can only spread like symptomatically. Like I think, for example, like rabies or something, they can only spread symptomatically, and then the host dies, right?
Yeah, that is correct. And also, other nuance to all this discussion that we are having is that you can have exactly the same parasite and the same genetic variant of that parasite, but depending on the host species or the host individual that same parasite is infecting, the reaction is going to be different. And you saw that with the COVID pandemic. And the opposite is true as well. Depending on the genetic variant of the parasite can end up dead or becoming the next pandemic, and anything in between. Okay? So it's a very complex dynamics, host-parasite interactions. Okay, and the reason we have that beautiful fish in this picture is because they live in more or less dark conditions, and they produce light in these white patches, but the light is not produced because of the tissue, the structure of the tissue, as it can happen in different materials, but it's produced by bacteria that live in that patch. Okay? So another type of symbiosis. Okay. So one thing that I hope comes out of this nice discussion that we are having is that prokaryotes or microbes are not only bad news. We humans have positive and negative effects. So nowadays, especially during the last decade, it has become very apparent that microbes are a welcome thing into human endeavors and human health. Okay? In terms of industry, we use pathogens to produce a lot of our food. I will mention some examples in a second. But then, we also need pathogens to be healthy. So the famous thing nowadays, especially during the last 5 years, is the micro what? Microbiome, okay? Which is true. Now, the microbiome has to be healthy. If you are just eating chips, fries, and burgers, you are going to get sick. Not only because you are not getting the nutrients, but because you are also feeding the wrong community of microbes in your digestive system. So you want to favor the right microbes in your digestive system. Now, the other important thing about the microbiome or microbes in general is that most of the cells that you have in your body are not your own eukaryotic cells. Most of you is prokaryotes, and prokaryotes that are beneficial for you. So you have a skin microbiome, also very important to keep it healthy. You have the digestive microbiome, very important to keep it healthy, okay? So all that adds to why it is important for us to study the diversity and the functioning of the microbes. So here are just very specific examples of some bacteria in our gut that are very important for synthesizing some of the important nutrients that we need for us to be healthy, okay? And in addition to that, some of the activities of that microbes in our body are going to produce signals that are very important for what? To activate genes that are going to be useful for us to do a proper metabolism. Okay, so some microbes that we want to have in our gut are going to tell genes to activate to absorb the appropriate nutrients. And in addition to that, some of the good microbes in our gut are a first line of defense as well when we are putting bugs through our food in our stomach. Why? Because they are going to produce antimicrobial chemicals, okay? So many important things about the microbiomes in order for us to be healthy and protected. And the only way for you to have those benefits is eating properly. I'm not saying to stop having dessert and burgers and fries or whatever you like of junk food, but just keep it to a minimum and make sure that most of your diet is healthy, okay? Now, pathogenic bacteria. So here is the bad news, right? Lots of those are going to produce human diseases. Now, one famous example is tuberculosis. Now, why tuberculosis? We were supposed to have it under control. Yeah, right. That's a wish we all have. There is no control in life. Okay, nothing is under control. So, tuberculosis, through our vaccination campaigns, we were very good at keeping it in check. We did not eradicate it, okay? And now, because we have allowed it, antimicrobial resistance, for many different reasons, social and biological reasons, now tuberculosis is back, and is back stronger than ever. It is producing a lot more infections, and now our combinations or cocktails of antibiotics are becoming useless. Okay, so you better keep healthy, eat healthy, because if you get exposed to tuberculosis, your immune system and your microbiomes are the best protection that you have, because if you get sick, if this bacteria manages to overcome your defenses, you are going to be in big trouble. Lyme disease that we discussed at length already. So it produces thousands of infections and many, some deaths, not many deaths, but still it can become a problem, okay? Now, how do pathogens produce the problems that we call symptoms and disease? the two main ways is by the production of toxins. One is exotoxins, meaning while the bug is in your system, is producing chemicals that are toxic to your system, and then you get sick, okay? One example is Vibrio cholera, which produces digestive disease, okay, when you eat contaminated food. But then we have endotoxins, which are these complex macromolecules like lipopolysaccharides, a combination of lipids and proteins. So they are part of the component or the structure of the prokaryote, of the bug. But once the prokaryote dies within the organism, or whatever source of potential contamination they are living in, they are going to disintegrate and they are going to release these chemical components. So those chemical components are the ones that are going to produce the food poisoning or any other diseases that we record. So two ways: exotoxins, meaning active production of chemicals that produce disease; endotoxins, so those chemicals that after the organism dies become poisonous to the host, okay? Now, we have already mentioned this, horizontal gene transfer in the three different versions. What are the three different versions of horizontal gene transfer that we learned today?
Transformation and
Transformation,
conjunction
conjunction,
and
transduction. Okay, three types. Which one is the classic one where we see the picture of the bacteria connecting through a tunnel? No? Conjunction. Okay? Which one is the one where the virus is basically acting as a mailman transferring information from one cell to the other? Transduction. Okay? Good. All right. So, this problem of producing bacteria that are not harmful and turning them into evil bacteria that produce disease can happen even within our microbiome, like E. coli, which is a famous component of our microbiome. Usually we have good E. coli in our digestive system, but if we expose our microbes to those microbes that have these plasmids or the genes that confer resistance, then they can become evil and produce disease, okay? So, antibiotic resistance basically has been recorded in history since we invented the first antibiotic, which was what? What was the first antibiotic that we used? Penicillin, okay? So, and if you remember your history, that antibiotic was developed by mistake, okay? Because some researcher trying to find something suddenly realized that the infections or the contamination in the petri dishes or cultures that it was using, it had these discs of things that where bacteria wouldn't grow. Okay, and that's where penicillin came from. But then as soon as we discovered penicillin, we started to see resistance. Here's a graph that is in your textbook of different, a list of antibiotics in here, when they were discovered, when we started to use in medicine, which is the black portion, and when we detected resistance. Okay, so you can see in here that sometimes some antibiotic, the famous penicillin, there were already resistant bacteria even before we started using it to treat bacterial infections. Okay? But it was a game changer at the time because it was the only antibiotic that we had, and it was very good to save the lives of many soldiers in the field. Okay? I have already mentioned this, but it's good for you to keep it in mind. So rapid reproduction is one of the keys for these bugs to develop a lot of resistance, because rapid reproduction means many more generations means more accumulation of novel mutations, but in addition to that, they also have what? Sexual reproduction, 3 different types. So they can transfer genetic information in many different ways, okay? So those are important things. Okay, I already mentioned this about tuberculosis, that now we have a strain that is highly lethal and highly resistant to most of our drugs. By 2017, more than 100 countries had confirmed cases of this resistant strain of tuberculosis. So it's all over the place. I mentioned this, there are few treatments available. Yet, of course, there is always hope, right? So there is a new class of antibiotics called malacidins. Okay, those malacidins, quote unquote, recently discovered, turn out to be our new weapon. Now we need to learn to not misuse it, otherwise, in no time there will be a resistant bacterial strain. So you can see here that when they were using these malacidins, when they use it in this experiment, when they use it right away after 24 hours, the bacterial infection was killed. And then the control, you can see when they were not using antibiotics, the difference in growth rate. So this is a new set of chemicals that can be very useful. Other ways that we have developed for medical treatment is the use of viruses, basically as vehicles to deliver the thing that we need in order to kill the infection, okay? So in particular, multi-drug resistance. So now virologists have discovered the code in order to make these virus vehicles very specific for delivery. So basically they have a GPS for them to reach the place where they are supposed to deliver the load that they have. Okay, so this is another active area of medical technology that is very hopeful in order to treat these things. For our own purposes, food industry, we use bacteria for fermentation. So we produce yogurt, we produce beer, wine, and many other foods. Now, other applications, and this is the workhorse in molecular biology labs, is PCR. Why we can do PCR routinely in our labs? Well, thanks to a bacterium. Why a bacterium, especially, or a prokaryote? Why a prokaryote? Because we are using the enzymes, okay, the polymerase enzymes that are the machinery, the molecular machinery to replicate the genetic material from an extremophile, okay? A prokaryote that lives under high temperatures, okay? Why is that important? Because during PCR, what we are doing is that we have this cycle where we increase the temperature to 94, 95 Celsius degrees to denaturate the double helix. So we open that. But at 94, most of the enzymes that we will have in our body would denaturate, so we would die, right? So 94 is fairly high. But for the polymerase enzyme of this prokaryote, 94 is just perfect living. So that's why we use it in molecular labs, and that derives again, that enzyme was derived from a prokaryote. Now the most famous one is the CRISPR-Cas9 system, which is a system that prokaryotes use as a defense mechanism against viruses. So it's like type of an immune system for bacteria to defend against viruses. But we hijack that system in order to use it to do a lot of genetic engineering, okay? Questions? More seconds. Okay. Yeah, correct answer is E. All right. This system that we just discussed, CRISPR-Cas9 system, has been very useful to do further study in in AIDS, in the HIV virus, and other applications that we have from prokaryotes is in the production of materials that we use for everyday things, like for example plastics. So there is this example of this bacteria that synthesizes this type of polymer known as PHA. And now we are basically farming this bacteria in order to produce these plastics that can be recycled. So those are environmentally friendly plastics, okay? Other ones are used to produce ethanol. So as you know, ethanol can be a fuel for many of our vehicles. Also, we have modified prokaryotes in order to do bioremediation work, especially after spills, petroleum spills. So all those bacteria are useful in order for us to clean our mess. Okay? Any questions? No? Okay, 10 more seconds. Okay, don't pack up just yet. We have some more minutes. I will start the next chapter just briefly. Okay. All right. Any questions before we move into the next chapter? Well, okay. So I'm just going to give you a brief intro to this chapter on protists. Most protists, or what we consider protists, are single cell, but what type of cell? Eukaryotic cells, okay? Now we're moving from prokaryotes to eukaryotes. But before we jump into complex organisms, we are going to deal with a huge diversity of organisms, all engulfed in the term protists, that are eukaryotes but are single cells. And as we have discussed earlier, they have been the product of endosymbiosis. Okay? So before we finish the lecture, let's make a summary. We already talked about endosymbiosis in previous lectures, and we talked about serial endosymbiosis. Okay? So if you remember a little bit about the history of life on the planet, what was the first endosymbiotic process? What was the primary endosymbiosis?
Just the involvement of the mitochondria and chloroplast. Yes?
Mitochondria only.
Oh.
Okay? First endosymbiotic process was the mitochondria. And why the mitochondria was important, especially at about 2.4 billion years ago.
To produce energy for the cell.
And in addition to that, what about the atmosphere?
It was aerobic.
Aerobic, okay? And oxygen was starting to increase, okay? So that's why mitochondria was very important, either as a free-living protobacterium or now as an endosymbiont for eukaryotic cells. Of course, eukaryotic cells didn't appear in the fossil record 2.5 billion years ago. They appeared much later, about 1.8 billion years ago, okay? Now, the other, in serial endosymbiosis, the second important process and key for the life on the planet was the plastids, okay? And what was then the second important endosymbiotic process or secondary endosymbiosis? Chloroplasts, okay? Which allowed for photosynthesis of eukaryotes, okay? So we will see that within the protist we have tertiary endosymbiosis. Okay, and some of the groups listed in here we will discuss in more detail during the next class. So let's take this question and then we can leave.